So I'd like to thank Damir for inviting me, and but I guess complain a little bit because it's always a humbling experience following Scott Diamond. But I'm going to dumb it down a little bit by necessity. I just arrived on a red eye, which is why my eyes are red. So, but with every last fiber, I will try and deliver this talk. I'm going to just tell us a story, not much modeling, a bunch of biological data, and focusing on a mechanism that we've really developed over the years, more knowledge on. It has to do with how neutrophils get to the site of inflammation, which is a really important process. We've learned a lot about this from experiments from nature. There's 3 leukocyte adhesion deficiency syndromes. LFA-1 is where integrins, there's a null mutation, and they can either express none or very few. And there's a LFA-2 where they don't have selectin, so they can't roll and stick. They don't have selectin ligands, that is. And then LFA-3, and LFA-3 is a very interesting deficiency. It's where at first they thought it was a GTPase, but more recently it's been discovered, even in the last few years, that it has to do with a molecule called kinlin that is a signaling molecule. So it's kind of interesting that these 3 pathways really define a lot of what we understand in terms of how neutrophils roll and stick and signal. I'm going to talk a little bit about the LFA-3 pathway. And it has to do with neutrophils getting to the site of injury. In all 3 of these syndromes, infections are prominent. Kids don't typically live beyond their adolescence, and they have very non-productive abscess where there's no real pus formation. And so these kids suffer from really bad gingival diseases. When I was at Baylor, had the opportunity to actually study these with some of the MDs. And the story I want to tell today focuses on integrin-mediated guidance. I guess it's sort of a good place for me to be sandwiched between Scott Diamond and Dan Hammer. Not good because you're not going to be impressed with my work after you hear theirs, but the fact that I'm talking about a calcium-mediated process of signaling. And it has to do with the so-called outside-in mechanosensing that is really just at the beginning of being understood. And we've also developed some microfluidics. Scott and I both come from Rice, where they had also developed some clunkier types of plastic devices with rubber O-rings, and about half of the time the experiments would fail. And these give us the ability to do, so 4 studies in one simple little PDMS disc, and so we can add blood, either whole blood or isolated neutrophils, have a negative pressure, and pull these guys across the substrate. And you can actually observe the kinetics of adhesion, much like Scott was talking about, except these are, like I say, bigger, dumber cells that roll. You can see even as they're rolling on this substrate of a cell line expressing E-selectin and ICAM1, and I'll talk about some of these molecules in a second, you can see they stick, and you can see they rapidly shape change after they stick. And this is just circling through again. So you can observe all these processes. You can obviously control the situation more than you can looking at it in vivo. And so I took the in vitro approach. Some of my other colleagues have taken the in vivo approach, and we tend to publish stuff in, Annals of Biomedical Engineering, and then they publish it in JCI, but such is the life of a bioengineer, right? The other advantage of in vitro studies is you can label these guys with fluorescent dyes, and so you can look at integrins and selectins. And what's been really interesting is looking at the dynamics. And even though certainly we understand something about single molecule on rates and off rates, what seems to dictate how neutrophils get to where they need to go so efficiently is they redistribute these adhesive molecules, and they link up to cytoplasmic proteins, cytoskeletal proteins on the inside. And that's also a theme of what I'm going to talk about today, is dynamics by which, in particular, integrins, not so much selectins, that's something I've studied in the past. We're looking a little bit more now at the integrin process, which is a little bit further down the line, and trying to understand how the integrins, which really are the last thing that the neutrophil is holding on to the endothelial cells with after they adhere through selectin. So when you have an inflammatory insult, the endothelial cells are activated through cytokines to produce, in a transcriptional manner, upregulate selectins. E-selectin seems to be the important one for any type of acute to chronic inflammation, and that's what we've focused on. And so these selectins enable neutrophils to roll. And as nature sometimes reuses molecules in different strange, beautiful ways, really, E-selectin also can signal into the cell and activate this second part of the process, which is simply integrins binding to ICAMs. I'm specifically talking about beta-2 integrins. In the platelet, Scott was talking about beta-1 integrins. So there's 4 different types of beta-2 integrins, these heterodimers that have alpha and beta subunits. And I'm talking mostly about the 2 that seem to dominate how neutrophils stick and migrate and transmigrate. And that's LFA-1 and MAC1, CD11A and CD11B, CD18. And like I say, the leukocyte adhesion deficient patients don't have a whole lot of, or sometimes no, beta-2 integrins. And so oftentimes what you'll see is they can still roll normally and sometimes even stick because there's a little bit of beta-1 integrin on neutrophils. But they don't seem to be able to do this process of migrate into the site of insult. And the transfer coefficient is quite amazing. We've been studying this process in mice, and mice circulate 100-fold fewer neutrophils in their blood than humans. We circulate like 5 million per mil. Mice circulate like 10 to the 5th per mil. But the same number get into the tissue, which is kind of amazing. So this transfer coefficient between the bone marrow producing neutrophils at a rate of about 1 million an hour, or 5 million an hour, and getting into the tissue at the site of insult is quite amazing, quite efficient. And the other thing that's really that I'm going to talk about is that integrins, as they're engaging or in some way interpreting their environment, their chemical and mechanical environment, to guide this process. And it's always been really interesting that neutrophils, as I'll show you in a minute, it's not at all a stochastic event. In fact, you can't really even model this as a stochastic event, I don't think, because when neutrophils, like I say, build up in tissue, they roll and stick within about a cell diameter or 2. And they end up at tricellular junctions, about 80 or 90% of them. Like I say, so they roll at 10 microns per second. They stick within only about 2 cell diameters, and they transmigrate within less than a minute to get out to where they need to go. And so this shows you what it looks like. This is in very low shear, and in very low shear it's even less efficient. But you can see this neutrophil with a classic pseudopod and uropod finding the site, and then when it becomes phase dark, it's migrating. I've never quite understood this process. Maybe somebody can talk about it. Basically, if you look at the viscosity of a neutrophil, it's like road tar, and it goes through a 100 nanometer pore in 30 seconds. Go figure that out. And when it does it, it looks like the viscosity of the cytoplasm is actually quite liquid. And so that's another foreshadow, because calcium probably has the ability to change the viscosity of cytoplasm pretty dramatically. And so because I only have 30 minutes, I'm not really going to dwell so much on the molecular anatomy, except to say the molecules I'm going to talk a little bit about, like I say, are the LFA-1, because that seems to be what's important for this mechanical guidance, that is, when it first rolls to a rest and then starts to shape change. And I'm going to talk about this molecule kinlin, which is the LAD3 phenotype, where it's missing the kinlin. And also I'm going to talk about a calcium channel that we've found to be really important in at least a trimolecular complex that enables integrins to sense their molecular and mechanical environment. They actually navigate using stress acting on these LFA-1 bonds through communicating with these cytoplasmic proteins that I'm going to talk about. So the question is, do integrins sense their molecular environment? Do they use that to navigate appropriately, like I say, within 2 cell diameters to transmigrate? And then how do they do this? And we're really only beginning to understand this. So this term mechanotaxis, a show of hands, how many people have heard of this term before? So mechanotaxis is a little bit like chemotaxis, because the same effect of getting a neutrophil close to a pore and transmigrating, except what has been discovered using experiments both in vitro, and this is a set of studies my grad student did recently. Instead of following a chemokine gradient, they actually use the directionality of the shear vector to travel in the correct direction. And we studied this by using a T junction in which we observed neutrophils, like I showed you, sticking and then migrating, and these are the trajectories of these cells, migrating, and then within about 10 minutes switching the direction and then seeing if they could then detect the shift in direction. And we looked at the dependence on one of the integrins, MAC1, and the dependence on the other integrin, LFA-1, so I'll cut to the chase. And LFA-1 seems to be the important molecule for detecting the shear direction. And I'll also say that this calcium channel, ORI1, that I just showed you is going to be important in my story, is also important because for the most part, you see that they're biased in a y direction if the shear is coming at 0 degrees. That is, they move perpendicular. Every once in a while, want to be carried down with the flow, but for the most part, they move perpendicular, even against the flow. And that bias to orient themselves perpendicular to the shear flow actually is lost when you simply knock out half of those calcium channels. And the way we quantified that is simply looking at the delta y over the delta x, and you can see that the total distance isn't all that different when you knock out that channel, but the ability to bias is significantly different. It's knocked down by about 60%. Coincidentally, I was collaborating with Paul Kubes up in Calgary, a really fine leukocyte immunologist, and he made this observation that another molecule that's downstream of the calcium channel, that's a GTPase molecule called VAV1, had a similar phenotype. And like I say, these immunologists are much smarter than us. They typically go right to the mouse and look at this. And this is a mouse microvasculature. And you can see this really the same process. And these guys are migrating, I should have said, on ICAM1. And a lot of what they're migrating here is on ICAM1 upregulated using TNF and a chemokine in this venule. And when you knock out VAV1, you can see that they basically get carried downstream. The percentage of neutrophils that crawl in the direction of flow is much higher, so they get pushed downstream. I guess nature has made this so is because if you're moving perpendicular and you're looking for the site at which you're going to transmigrate, it's much more efficient if you're doing that circumferentially in a vessel, right? And so somehow the integrins, because if you knock out the integrin, they don't do this, mediate this process. And so let me talk a little bit about the molecular dynamics of CD18. And Tim Springer and Ronan Allen and others have done a lot to really show how it is that going from primary amino acid sequence to tertiary structure, how integrins work, and it's really some beautiful structural biology. And in the presence of calcium, you can stabilize integrins, and this is a scanning EM, in this bent or genuflected state. And when the cells become activated from the inside out through, say, a chemokine, it opens up and it actually spreads the alpha and beta subunits. And this is what's associated with high affinity. There's also been an intermediate affinity state that's been observed called the extended conformation. And LFA-1 seems to use this to decelerate a neutrophil, this extended conformation, before it achieves the extended conformation and mediates neutrophil arrest. And we have some tools in our pharmacopoeia to mess around with the integrin structure and then look at function. So one is lovastatin, just simply by a weird coincidence of nature. This statin recognizes the I-domain allosteric site and locks this into a bent configuration. We can identify this configuration because we could look at soluble ICAM1 binding to measure its affinity, or more phenotypically look at when neutrophils roll to arrest. We have antibodies that recognize only the open state, so that gives us a measure, and I'm going to use that as a tool in a minute to show you the dynamics of the excursion from low to high affinity as a neutrophil arrests and migrates. And then collaborating with some companies, Bert from Behringer and IC 487475. I just started calling it Amy because Amy used to send it to me, and the papers that we've written with it, we call it Amy. You can see how, a bioengineer deals with biochemistry, right? This is able to stabilize the extended conformation. So there's a bunch of really cool tools out there, allosteric tools, in order to stabilize an integrin conformation. And one thing that we learned was that neutrophils, when they do roll and stick through selectins and integrins, that these receptors redistribute in very specific ways. There's nothing really chaotic about this. The high affinity integrin finds its way down to the substrate. And if you use confocal slicing, you can see that there's much less of the integrin at the point that they're arrested as you move up. L-selectin, one of the molecules that enables this guy, when it rolls on E-selectin, to reach this high affinity state, is actually in a different place. So there's a lot of sort of action at a distance that occurs during signaling of neutrophils to arrest. And this simply shows the quantitation of that. So what responses downstream from this shift in integrin affinity and avidity? And when I say avidity, I mean clustering of high affinity sites that I just showed you, are important in this process of mechanotaxis and transmigration. So the first step, as I said, is E-selectin, and you saw that L-selectin was sort of more closer to the center, or at least a couple of micron up, because these guys form these beautiful little microvilli. The neutrophils also engage chemokines, which also signal to the cell. And within seconds, really, as I said, just a couple of cell diameters, you can observe these high affinity clusters of integrins at the substrate. And this has been observed not only by me, by others, and it's been known that this micro clustering and then macro clustering is important for the process of neutrophil arrest and then also adhesion strengthening. So if the neutrophil doesn't achieve these small clusters, the shear stress will pull them off of the substrate. And so I'm going to describe some experiments in which we've used some of these molecules to either stabilize a low affinity, block the high affinity with an antibody, or the extended conformation, and see what the effect is on the kinetics. And you can see, if you look at rolling velocity, delta x, delta t, as a function of time, normally a neutrophil rolling on E-selectin has a very nice smooth, about 4 micron per second rolling velocity. If you block the intermediate state, it increases the velocity somewhat, and it's a bit more chaotic, but they're still able to sort of, they're still able to roll, because E-selectin is mediating the rolling. If you block that, then simply they roll in a very chaotic fashion. So this shows that LFA-1 conformation, because this is specific for LFA-1, is important in the deceleration and also in the smooth rolling. But it also gave us the first inclination that the conformation of the integrin was important in how the neutrophil is interpreting its shear environment. And this is shown quite beautifully here. I'm going to show all three, both LFA-1 and MAC1 in dynamic studies in which the neutrophil has just arrested, and then they start to polarize, which is the next process after they arrest, adhesion strengthen, and start to migrate just before they transmigrate. MAC1, you can see, forms a nice little cap towards the end, towards the uropod. And I think Dan Hammers explained nicely that a lot of the traction is really being generated in the rear of the cell. He might talk about that. LFA-1 has a very different conformation. You do see it forming at the rear of the neutrophil, but we always would see, wherever a new pseudopod was being sent out, that small clusters of LFA-1 would be sort of, in a sense, guiding the neutrophil. Didn't quite understand how that was working. The question we asked was, is that high affinity, or is that just LFA-1 binding? And if you use that 327C, you can see it's even more clear that there's high affinity sites at the rear and high affinity sites just at the point that a neutrophil is interpreting where it is and sending out a pseudopod. If, again, you use that small molecule inhibitor and simply they can still stick, there's still ICAM1 down here, and they're still sticking to ICAM1 through the integrin subunits, but they can't seem to interpret the mechanical environment and send out pseudopods and migrate. So I haven't shown you any modeling, because we didn't really model this process, but we have observed this really interesting phenomena. And when you start to look at the molecular pathways that are involved in this is really what's kept me from modeling this process, because when a neutrophil is attached, engaging integrins, sensing chemokines, still receiving some of those selectin signals, it's getting at least this many inputs and activating a whole slew of processes. And I thought about what is the common molecule that all three of these processes, that is CXCR activating diacylglycerol and calcium release, Dan's going to talk about this in T cells, integrins are not only shifting to high affinity but clustering and engaging their ligand, and selectins are also mediating a signaling process that eventually activates MAP kinase and downstream will activate the integrin in and of itself. All three of these, a common theme is calcium flux. And so it made sense to measure calcium flux, almost as if the calcium flux was like the RPM of this cell. And so we did this by loading cells with a calcium-sensitive dye, fluor-4. It actually, if you look at the ratio, it gives you a measure of how much calcium is being produced in the cell in a very quantitative fashion, much like what Scott Diamond talked about, the integrated signal. But we were interested in understanding how calcium was being modulated from the time it touches down, as it rolls, and as it arrests. And then Fura2 is a much more rapidly emitting fluorophore that doesn't bleach as fast. And this gave us, once we knew what the correlation was between concentration, we simply used Fura2, where you didn't need to use a ratio. And we wanted to deconvolve the calcium signal in the context of all of the responses that, all of the signals that a neutrophil is getting as it rolls and stuck and migrated. And so that looks like this. We have neutrophils rolling on a substrate, an L-cell substrate expressing E-selectin and ICAM1 in physiological concentrations. And you can see right there, they're already starting to glow green. Right here, we infused IL-8. And within, very rapidly, the cells not only arrest, but then start to flux calcium. So there's definitely calcium either being released from the inside of the cell or also being taken in from the outside of the cell in response to these signaling events. So does integrin engagement cooperate with this calcium influx to activate the neutrophil and inform it of the fact that it's binding ligand and it needs to then change shape and migrate? And in order to do that, I mean, it sounds simple, but in order to do that, you really need to start to understand what the signaling components are. That is, we're getting a G protein coupled signaling event that activates the endoplasmic reticulum through diacylglycerol and phospholipases. We have a selectin-mediated calcium event that might also activate calcium release. It's not totally understood what pathway it goes through. And then there's also calcium channels. There's TRP channels and there's RI1, both important for neutrophil function. And so the real question is, what is the role of calcium in signaling integrin engagement and also integrin recycling and rebinding as a cell migrates? Interestingly, at the time that we took up these studies only a couple of years ago, almost no studies on neutrophil function were done under shear stress, under the normal conditions in which they roll and stick. Almost all of them were done in cuvette or test tube studies, because it's much easier to measure enough signal and even dynamics using flow cytometry or spectrophotometry. These are a little bit tedious. You're tracking a single neutrophil, like Scott Diamond did with the platelet, and you're doing that over time. And the first question we ask, well, what is the dose dependence of the calcium flux as a neutrophil rolls and sticks under different concentrations of IL-8? And you can see it's a very beautiful relationship that at very low concentrations that just start to trigger arrest, you can almost see no calcium flux. About 80% of these guys never will stick tightly. As you increase, you get more and more cells that stick tightly. And if you then reinterpret this data in terms of a dose curve, we observe something that was really interesting. So you see about the same dose dependence as you see with other measures of neutrophil function, like cell arrest or shape change, or even integrin upregulation. But the really interesting thing was if you did these experiments by just letting neutrophils sit statically on E-selectin and ICAM1, you saw nearly a 500-fold, well, first of all, the baseline was higher. So rolling on the selectin itself increased some of that calcium signal. But the other thing is that it's much more sensitive when a neutrophil is rolling on E-selectin and ICAM1 to a dose of IL-8. Like I say, like 500-fold more sensitive. So what is attributing? What is attributing this? We looked at it a little more carefully in terms of the dynamics of rolling and sticking. And what we saw was, so what we did was we put a clock on the number of neutrophils that roll, decelerate to arrest, which is blue. So that's on this side, 90%. The number of neutrophils that then transition to polarization, as I showed, and then also the calcium flux curve. And you can see that these guys are actually very sequential. That is, it's only after you get some calcium flux, about 25 to 50 nanomolar. It's only after the neutrophil really comes to arrest that you start to see the calcium flux build up, which gave us the hint that maybe integrins were assisting in the calcium flux that then enabled the cell to undergo these gross cytoskeletal polarizations. But we had to prove that to ourselves. And the way we did that was to do interventions. There's lots of pharmacological tools out there that enable you to either chelate the calcium inside the cell, to block the channel itself using APB, a pharmacologic agent, to inhibit the phospholipase C, which would go through the G protein coupled receptor, or to stimulate calcium flux altogether with an ionophore. And those are the interventions we did, and these are the curves that we got. So if you don't have calcium, you get very erratic cells that will arrest, but it's not very efficient, and most people know that. Neutrophils need calcium on the inside to do their thing, even more so than on the outside. They need calcium to roll because selectins need calcium. If you use APB, you could see about half the cells will still arrest, and not very tightly, but obviously you block a lot of the calcium flux that we got by blocking this extracellular channel. So that told us that some of the calcium is coming from the outside of the cell in very rapidly in mediating the response. We proved that to ourselves by using an ionophore, and this was kind of what blew me away and sort of made the lights go on, is that even in the absence of IL-8 or any other stimulus, ionophore will jumpstart the process of arrest, calcium flux, and cell polarization. So calcium in and of itself on the inside of the cell can mediate all of these responses, not in a very synchronous fashion like we saw, like you typically see. And if you block that extracellular calcium with ABB, even if you activate with ionophore and you get some calcium flux, you still don't get that synchrony. So the process of rolling arrest and then sensing that shear is really what enables the cell to flux that calcium. And that was proven in these studies where we went from low shear to high shear. We looked at calcium flux, and we also then quantified polarization, which requires that calcium. And you can see here that that shear itself can flux that calcium, just like the ionophore did. We then also looked at calcium entry in the context of using, and you can't see this because I've covered up the slide, pardon me here. So, kind of jump starting this. So what we did was we used thapsigargan. Thapsigargan will basically deplete all the IP3 calciosomes in the ER. It will enable a cell to only respond to cell coming from the extracellular receptor. Obviously, we're really interested in looking at RI1, as I said. And then you could look at function, which is what I'm going to show you now. And so these experiments are kind of fancy, because at this point we got ourselves a turf microscope. And so you're only seeing calcium within the contact region of the cell, within 100 nanometers of contact, because that's the only way the fluorophore can be excited with this on-phos laser. And you can see only after a cell tethers do you see the calcium flux. So it is the tethering that triggers the calcium flux, and it is at the site of contact, which was sort of the next light that went on our head. That really it's probably integrin engagement as well as that calcium channel that's enabling cells to decelerate. And in fact, thapsigargan, just like I said, showed the ionophore itself, in an even more synchronous fashion, can activate calcium flux and also polarization of the cell. It was about that time that this paper came out, showing that, like LAD1, 2, and 3, there was another immunodeficiency identified in an RI1 mutation. In fact, these were discovered in B cells and subsequently T cells by Anjana Rao and other folks at Stanford that reported that they have a deficiency in producing an immune synapse. and producing sort of an adaptive response. And subsequently, in the data I'm going to show you, we showed that there was also an innate immune defect. The other paper that came out right around that time was the LAD3, where Kinlin-3 was important for integrin activation, clustering, and high affinity. So it was these two papers that really made us think that maybe these molecules were working together. And that's where I'm going to jump to the punchline a little bit before I show you that data. Kinlin is known to associate with the cytoplasmic domain of an integrin. It's not calcium dependent, as I'll show you. Talin works with Kinlin to form clusters and strengthen the arrest process. The question is, where does ORI1 come in this process? And the question is, what's the mechanism by which Kinlin and some other adaptive proteins might cooperate to regulate where and when the calcium flux occurs, as I've just showed you experimentally that it does. And to do these experiments, my graduate student Neha Dixit came up with a really elegant means of avoiding G protein coupled receptor activation or anything else other than simply ligating an integrin and putting stress on it. And that's using these allosteric antibodies that we got a hold of. This antibody actually can induce the high affinity conformation by binding. This antibody from Tim Springer binds and actually stabilizes a low affinity. Both of these antibodies bind with nanomolar affinity and will hold the cell as you shear it. So we're able to sort of hold the cell by either high affinity or low affinity, induce stress, and then look at calcium and also look at Kinlin. And that's the last experiment I'll describe here. In these experiments, we looked, first of all, as ORI1 and LFA-1 co-localizing under conditions in which we induced high affinity. And only under conditions of high affinity did you see the calcium channel align with LFA-1, not under conditions that they're being sheared. So tension is going across the bond, but not in a high affinity conformation. That was really interesting. The other thing that we saw was that high affinity and tension and shear stress was required both for Kinlin to build up at these high affinity sites and for the high affinity sites to be expressed. So Kinlin and ORI1 are showing up at the same place when the receptor is under tension and under high affinity. We knocked out Kinlin and saw that neutrophils don't shear strengthen during arrest, and not true for Talin. You see a much more mild phenotype with Talin. So Kinlin is absolutely required, even though Talin is what's been thought to be the molecule associated with strengthening of these bonds. And then the other thing that we noticed was only under conditions in which you knocked out Kinlin do you not get these larger clustering of high affinity integrin. With Talin or under control conditions when you stabilize high affinity states, did you get these sort of micro to macro clusters that are required for shear strengthening? So Kinlin is calcium independent to build up at those sites. Talin is calcium dependent, and that's been known. So Kinlin becomes a very sort of attractive candidate for a molecule that's calcium independent to link up to the cytoplasmic domain and transmit force and in some way link up with the ORI1. In the ORI1 knockout, you still got Kinlin binding at those high affinity sites. And in the last experiments we did, we simply looked to see if these molecules were physically associating and under what conditions that was true. And only under conditions that you had a high affinity and that you had shear stress, and if you immunoprecipitated with LFA-1, you could see that you would pull down both Kinlin and ORI1. So you can actually pull down a complex that's covalently or non-covalently strong enough to withstand a Western blot. And the last data I'll show is that, like the cooperativity I showed you with ILA concentration and calcium flux, Kinlin is absolutely required for that. So in neutrophils or HL60 cells where we've knocked down Kinlin, you don't see that amplification, that normal amplification you see where a neutrophil is actually activating integrin and amplifying the ability to produce calcium locally. So the model that we have at this point is a little bit different than the model that has been reported by Feigeson and Alon in T cells, where they looked really closely also at the dependence of Kinlin. So T cells and neutrophils both require Kinlin for outside-in signaling, T cells don't seem to need kinlin for adhesion strengthening under the conditions where calcium is required, and neutrophils absolutely do. So we're not exactly sure if kinlin is the mechanosensor that enables an LFA-1 to inform the inside of the cell that calcium flux should be locally occurring at this site, and therefore reinforcing association with the actin cytoskeleton. The other question we have is how local calcium regulates the cytoskeleton and mechanotaxis. What are the other molecules that are associating, like VAV1, CDC42, and other GTPases? And another interesting question in this regard is, can this pathway be exploited? Can we get small molecules that will really focus their effort on blocking this kinlin association and therefore neutrophil arrest, but not really affect other leukocytes like T cells and B cells? Because neutrophils and T cells and B cells seem to be different in their ability to localize ORI1 at these sites. I'll stop there and I'll acknowledge the people that did this work. Uli Schaff, who made a lot of the microfluidics and did all of the calcium, the initial calcium work. Neha, who picked it up from her. And Chad, who gave me those beautiful integrin real-time images of high affinity as neutrophils migrate. And of course, my funding sources. Thanks, and I'll take any questions.